All right, hi friends, welcome back. Today begins our quest to understand biomolecular structure and function. This is going to be a shift away from new reactions all the time. We'll learn some new reactions, some new synthetic strategies, but for the most part, our focus for the next whole unit is going to be on the relationship between the structure and function of biomolecules. We want to set the stage for you to understand when you get to 481 biochemical systems. And to do that, you need a strong foundation in biomolecular structure and function. We want to see the relationship between how the structure of a biomolecule is related to its function. We'll talk about a lot of that today. We'll be flexible with the schedule here. Today we begin amino acids, peptides, and proteins. I schedule about 3 class periods on that. I have never, ever successfully held it to 3 class periods, in part because peptide and protein chemistry is what I do in my research, and so I just sort of geek out on it, and I can't resist. So you will be the beneficiaries, or actually guinea pigs, or anyway, what I mean is you'll have to suffer along with me as I geek out on some of this stuff that I think is really cool. But the good news is what you learn here is going to make the first part of Chem 481 a lot easier. So let's dive into amino acids, peptides, and proteins. I'll draw for you an amino acid. We generally call these alpha amino acids because the carbon that is alpha to the carboxylic acid also has an amino group on it. I'm going to draw this amino group in its ammonium form so that I can show you what its pKa is. And then here is a side chain that is a dash, and a wedge. The wedge is a proton, which is often implied but not shown. This alpha amino group in its current ammonium form has a pKa that's around 9, meaning at physiological pH that proton is still on. The amino group of most amino acids in your cells should be positively charged. The carboxylic acid group, in contrast, has a pKa of around 2. And so at physiological pH, it should actually be in its negatively charged carboxylate form. So this is the form an amino acid would tend to adopt at physiological pH. Note that the side chains often have groups that can also be protonated and have characteristic pKa's. We will learn those, just not now. We're just going to be general about the side chain. The 20 amino acids that are present in biology all have L stereochemistry. What does that mean? Well, L and D were the old-time ways of talking about chirality and stereochemistry before a lot of modern analytical techniques were used and before we had the priority rules that allow us to assign R and S for stereocenters. And it was difficult to assign L and D. You did it by analogy to known compounds, and the history there is not that important for us today. But we still call them L amino acids. And the way that I remember it is when I draw the amino acid this way, with the ammonium group on the left, the carboxylic acid group on the right, and the alpha carbon at a valley position, the side chain should always be a dash and not a wedge. Now, of course, if I flip it, move it around, I've got to adjust accordingly. But that's what I've got in my mind is sort of the default stereochemistry for amino acids. Most amino acids, well, I will say all except cysteine, which you will learn about cysteine in a little bit, are S at the alpha carbon. Cysteine is R at the alpha carbon because of a technicality. Cysteine has a sulfur side chain, which causes the side chain to win a priority rule. But you can see why this would be S. Nitrogen is priority 1, carbonyl carbon is priority 2. The side chain, if it were, say, a methyl group, would be priority 3. 1 to 2 to 3, that's clockwise, but proton is up, so it's going to be S. So you'll need to remember that because you'll need to recognize an L amino acid that would be found in a protein from a D amino acid that wouldn't. Okay? There are some D amino acids in biology, but not many. Okay. Questions about stereochemistry of amino acids? All right. Let's now show what an amino acid looks like in the context of a larger peptide or a protein. So the term peptide is basically a polymer of amino acids. And they are linked together via amide bonds between the nitrogen of one amino acid and the carboxylic acid of another. This, for example, is a tripeptide. That's going to be a peptide with 3 amino acids in it. So here's the first amino acid, side chain. Here's the second amino acid with its side chain, and then here's the third amino acid. And I've drawn the ammonium group on this side and the carboxylate group in this side in their protonation states that they would exist in at physiological pH. Now right away you can see that peptides have directionality, meaning one end is different from the other. There's an ammonium end and a carboxylate end, a positively charged end and a negatively charged end. We call this the amino terminus, where terminus is just a more complicated word for end, but there you go, the N terminus. Probably don't say N end because it's awkward to say. This is the carboxy terminus, and we'll also call it the C terminus. And anytime you're thinking about a peptide, you need to get oriented towards the N terminus and the C terminus. That is, for this amino acid in the middle, the one over here is on its N terminal side, and the one over here is on its C terminal side. So we'll use N terminus and C terminus to keep us oriented. One of the ways you can tell or work your way through the directionality of peptides is to just notice that you've got alternating atoms in the backbone. You have a nitrogen, an alpha carbon, and a carbonyl carbon, then another nitrogen, an alpha carbon, and a carbonyl carbon. Over here, a nitrogen, an alpha carbon, and a carbonyl carbon. That establishes the directionality, N alpha C, and so on. Okay, that's important, N to C. And you can see that they have amino acids at the N terminus have a different environment than amino acids at the C terminus. For example, this N terminal amino acid that I'm highlighting in orange has a positive charge, whereas the C terminal amino acid that I'm highlighting in green has a negative charge, whereas the one in the middle doesn't have a charge on either side. Okay, so there are distinct environments. In a peptide that may be hundreds of amino acids long, most amino acids have this typical environment where they are internal. That is, they're not at the N terminus, they're not at the C terminus, they don't have a charge on the left or the right, they're just in the middle. It's these two amino acids at either end that experience this sort of unique charged environment. You'll notice I've highlighted each amino acid differently. It's important to be able to look at a peptide and recognize individual amino acid subunits. The way to do that is to identify the amide bonds, which I'm highlighting with these gray squiggly lines. The amide bonds are connections between the carbonyl carbon of one amino acid and then the amide nitrogen of the next amino acid. And we divide them up this way because that's actually how biology synthesizes them. It takes an amino acid, carbonyl carbon, alpha carbon, and nitrogen, and then it brings in another one and makes the amide bond. A synonym for amide bond is peptide bond. So you'll hear people use those two terms interchangeably. amide bond, and peptide bond. And the amide bond, as you know, has some interesting properties. You would expect the carbonyl carbon to be sp2 hybridized, trigonal planar. You also expect the nitrogen to be sp2 hybridized, trigonal planar. The reason you expect that is because this nitrogen has a lone pair set of electrons on it that are in resonance or conjugation with the carbonyl carbon. To take advantage of that, the nitrogen needs to be sp2 hybridized so that the lone pair can be in a p orbital where it can engage in conjugation with the carbon-oxygen pi bond. Okay? And then the alpha carbon, as you notice, is sp3 hybridized, tetrahedral. So in a peptide, you've got this alternating sp2 atom, sp3 atom, then sp2 atom, again and again and again. And that has some interesting implications for the shape that a peptide can adopt. Couple other things, the R groups, as I've already alluded to, are called side chains. And then the N, alpha, and C, which I'm sort of also highlighting in gray, that's called the backbone. So we'll talk about backbone atoms versus side chain atoms. Really, the carbonyl oxygens get counted as backbone atoms as well. So backbone atoms versus side chain atoms. That's all vocabulary. Any questions you have about that? Okay, yes?
So because the lone pair on the nitrogen is in resonance, it's not going to pick up another proton, so it can...
Yeah, that's right. So because the lone pair on the nitrogen is involved in resonance, the backbone nitrogen in the amide bond isn't basic. It doesn't pick up a proton. There's no additional protons. So you never need to worry about whether the amide nitrogen is going to get protonated at physiological pH. Basically, its pK, if it did, its pKa would be so low, ouch. Okay, that was weird, clumsy. Watch when I'm gesturing wildly. See if we can knock this over. All right, ow. Sad. It's fine, I'm fine, shake it off. Anyway, its pK would be so low that it would lose its proton, yeah. Okay, sorry, what else? Yeah, go ahead.
For each amino acid, does the R group flip stereochemistry?
For each amino acid, does the R group flip stereochemistry? No, but it looks like it has. The reason all of these side chains have the same L configuration, the reason this one is a wedge, whereas this one was a dash, is because in this drawing, nitrogen's on the left, carbon's on the right, alpha carbon's at a valley position, the side chain is a wedge. Here we got the same situation, nitrogen on the left, carbon on the right, but we're at a peak position, and so it would have, that amino acid would have had to flip over. So yeah, when you draw a peptide out in this zigzag shape, we call this a sort of a fully extended backbone, you'll see that the side chains will alternate whether they're dashes or wedges. Understand though, if you assigned R and S at each of those stereocenters, they're all the same chirality, except for cysteine, which is R on a technicality. But yeah, what else? Go ahead.
So all except cysteine are what at the alpha carbon?
All except cysteine at the alpha carbon have S configuration. And cysteine only is R, not because it has a different shape, but because it has a sulfur in its side chain. All of them have the same L amino acid configuration. Okay, others? All right, so now what we're going to do is we're going to zoom in on a peptide in a tripeptide segment in an existing protein, and we're going to talk about the conformations that can be adopted about the three types of bonds that are present in the peptide backbone. Here are the bonds I'm talking about. There's a bond between the sp2 hybridized carbonyl carbon and the amide nitrogen. Then there's a bond between the sp2 amide nitrogen and the alpha carbon. And then there's a bond between the alpha carbon and the carbonyl carbon. Those are the three characteristic bonds that any individual amino acid has. And we're going to think about conformations, that is, rotation around those bonds, what kinds of conformations are best. Some of this is going to involve digging deep back to 351, where you learned about Newman projections, which you hoped you'd never encounter again, but today is the day in which you do, and I'm not sorry. So a couple of things, because it's kind of fun, all right? So you remember that when we drew a Newman projection for an sp3 hybridized, a bond between two sp3 hybridized carbons, say for ethane. Do you remember drawing something like this? You remember that the cylinder represents the bond that we're looking down, that the carbon in the front you can see, the one in the back you can't see, it's on the other side of that circle cylinder kind of thing. And then you have CH bonds here and in the back. You know that those carbons are really sp3 hybridized, so those bond angles are really 109 degrees, even though from our perspective here, looking at it straight on, it looks like it's 120. And you've learned that this is a staggered conformation, and you've learned that staggered is generally better than eclipsed, right? Okay. Now, what do you think an sp2 carbon looks like from the side? An sp2 carbon's trigonal planar. So supposing I made the carbon in the front sp2 hybridized, what's that going to look like? It should look kind of like, I mean, we're making an imaginary molecule now, but it should look kind of like this, right? It's hard to draw. An sp2 carbon looks like that, flat, right? So you can see the problem now looking down a bond between an sp2 carbon and an sp3 carbon. We're no longer going to be able to get an exclusively staggered conformation. Do you see that one of these groups is staggered between the two groups in the back? The one in the front, though, is eclipsed. So there's going to be this sort of unsettled trade-off between what the best conformation is. And there may be multiple options that are allowed, and we're going to be considering those options, okay? So what we're going to do, and I've prepared some slides that take us through this with an actual protein structure that we have the three-dimensional coordinates for. You won't be able to copy it all down, but I'll publish them in the notes. Some of the Newman projections you can draw, and that would be useful for you to do. What we're basically going to do is look down each of these bonds in red and talk about the conformations that are adopted by each bond. So we're going to start with the amide bond, the bond between carbon and nitrogen. Here's the same tripeptide I just basically drew for you. Squiggly lines here indicate that this tripeptide exists in the context of a larger protein. There's amino acids on the N-terminal side of the orange one. There's amino acids on the C-terminal side of the green one. Also, I've labeled the backbone atoms N, alpha, and C for the orange, purple, and green amino acids respectively. It's not a huge deal, but in peptide chemistry, we often use the lowercase letter I to indicate our starting point for counting. So if we want to start here, this is the I position amino acid. This is I plus 1. That's I plus 2. You can go I minus 1 and I minus 2 as well, just a way to talk about relative positions of amino acids. These three amino acids I've actually taken from a protein that is alpha helical. This is a helix, a spiral pattern in one peptide, and there are three such helices that interact together to form what we call a helix bundle or a coiled coil. We'll learn more about that later, but not today. Today we're simply going to zoom in on the orange, purple, and green amino acids. Here's a little bit bigger. You can see the orange one at the I position, the purple one at I plus 1, the green one at I plus 2. And we're going to, one by one, look down the amide bond, the N to alpha bond, and the alpha to C bond. And we're going to assign a dihedral angle that describes the conformation adopted by each of these bonds. For the amide bond, that will be called omega. For the N to alpha bond, that will be called the Greek letter phi. And for the alpha to carbonyl bond, that'll be called the Greek letter psi, which I remember is called psi because it looks like the little sword that Raphael the Ninja Turtle used to have. Yeah, some of you know that. That was big when I was a kid. All right. Okay, so let's first talk about omega, the angle between the carbonyl carbon and the amide nitrogen. So here is the view from the actual 3D coordinates of this protein that we're zoomed in on. In these drawings, red atoms are oxygen, blue atoms are nitrogen, white atoms are, or sticks are hydrogens, and then carbons are colored based on which amino acid we're looking at, the orange, the purple, and the green. So we're looking at the bond, the amide bond between C and N of the orange amino acid to the purple amino acid. Here are our evil Anakin Sith eyes shouting, "I hate you" at Obi-Wan or at me. And you're looking down the carbon-nitrogen bond. And we're going to be looking especially at the relationship between the gray bond between carbonyl carbon and alpha carbon in the front, and then the bond between nitrogen and alpha carbon in the back. So here's the view. In here I've drawn the Newman projection. Here's the actual view taken from the image of the protein. In the front of the cylinder, you have the carbonyl carbon. Here's the carbon-oxygen pi bond going up, and the carbon-alpha carbon bond going down. That's where you see that planar sp2 hybridized structure. In the back, where we can't see it, is the amide nitrogen on the back side of that cylinder. The alpha carbon is here, pointing upward, and then the alpha proton, the amide proton, the NH bond is pointing downward. In this conformation, the NH bond is in the back is eclipsed with the C alpha bond in the front, and the CO bond in the front is eclipsed with the N alpha bond in the back. Okay, so that's what it looks like to look at an sp2 carbon on edge. Go ahead.
I was just wondering why the carbon behind the CO carbon is like planar instead of sp3.
So you're wondering why this nitrogen is planar instead of sp3? Yeah, the reason is the lone pair on that nitrogen is participating in resonance with the carbon-oxygen pi bond, so it has to be sp2 hybridized, so the lone pair can be in a p orbital where it could be conjugated with that carbon-oxygen pi bond. Okay, good. Now, we're going to define omega as the angle in this Newman projection between the orange alpha carbon in the front and the purple alpha carbon in the back. Here, that's 180 degrees. You can imagine another option where you would rotate around that amide bond, but such that the alpha carbons would now be eclipsed, and then the carbonyl oxygen and the hydrogen would be eclipsed with each other. If that were true, we'd call omega equals 0. Any intermediate geometry between 180 and 0 requires that amide nitrogen to rotate out and be perpendicular to the carbonyl carbon. That breaks conjugation, breaks resonance, and isn't favorable. And so you'll find that rotation around amide bonds is slow. It's possible, but it's slow because you have to break resonance and conjugation to do it. Here's a view of two different conformations of an amide bond from the side, alpha carbons on either side. This is what an omega equals 180 bond looks like. You can see that the alpha carbons are trans to each other. Here is what an omega equals 0 bond looks like. Here the alpha carbons are cis to each other. In general, trans is more favorable, or omega equals 180 is more favorable than cis, or omega equals 0, because the alpha carbons have steric clashes when they're both cis to each other. There are times in proteins where omega needs to be cis, and that's fine, but it's more common to have omega equal 180. All right, so what do you need to know about omega? That amide bonds are planar, and that omega has two preferred values, 0 and 180, but 180 is better than 0 because of sterics. Okay, questions on that? Okay, now let's do the fun one. Phi is the angle between, phi refers to the rotation around the bond between the amide nitrogen, which is sp2 in the front, and then the alpha carbon of the purple amino acid, which is sp3 in the back. Okay, so here's our sithis, that's the bond we're looking down. Remember that the nitrogen in the front will be attached to only two things, the carbonyl carbon and the amide proton, whereas the alpha carbon in the back will be attached to three things, the proton, the carbonyl carbon, and the side chain. So here's what the Newman projection looks like if I were to draw it, and then here is the view from the structure itself. You can see the nitrogen in the front of the cylinder, that's this blue nitrogen here. It's bonded to the carbonyl carbon here, straight up, the orange carbonyl carbon, and then the amide proton here in the front. That's an sp2 trigonal planar nitrogen. In the back, you can't see the alpha carbon, it's on the reverse of that cylinder, but you can see the alpha to proton bond here, the alpha to carbonyl carbon bond here, and then the side chain, which is here eclipsed with the NH bond in the front. We define the Greek letter phi as the angle between the two carbonyl carbons. In general, if you forget what omega and phi and ultimately psi mean, it's always the angle between the backbone atoms. So carbonyl carbon in the front and carbonyl carbon in the back. In this alpha helix, for this amino acid, the purple amino acid, phi equals minus 57. Now, you're never going to be measuring phi. And you may have the question, what makes phi equals minus 57 different from phi equals plus 57? Short answer is, if phi were plus 57, this carbonyl carbon would be over here. So it's sort of a clockwise versus counterclockwise thing. Really, it comes from vector math, and nobody does that in their heads anymore. You just ask a computer to do it. So I'm never going to be asking you, is this dihedral angle phi positive or negative? Just understand that the math takes care of that. But in this alpha helix, phi is minus 57 for this purple amino acid. Okay, are there questions about that? Yeah.
What's the angle for W?
For omega, W, the angle is either 180 or 0. So you can see sort of, did I see another hand? Sorry, yeah.
Of this diagram, you want us to take out that phi is negative 57.
Well, I want you to know what the definition of phi is.
Okay.
And the fact that it's minus 57 will become important a little bit later.
Perfect.
Yep. You can see the problem we've sort of got, right, with this sp2 atom in the front and an sp3 in the back. We can get the carbonyl staggered away from this carbonyl in the back, but then the side chain's eclipsed with the NH. So there's trade-offs in terms of sterics. It's more complicated than that because there's other stuff going on that you can't see. In 351, you learn to sort of think about keeping the biggest stuff the farthest apart when thinking about a bond between two sp3 carbons. But you didn't often consider additional atoms beyond the ones that are attached to the bond in question. For example, we might only focus on the carbonyl carbon, the proton in the front, and then the proton, the side chain, and the carbonyl carbon in the back. It'll turn out, I'll show you in just a second though, that things out further beyond those carbonyl carbons can start to bump into each other and have steric problems. So you'll see that unlike omega, which only had two values that were allowed of 180 and 0, phi can adopt a lot of values, and some of them are going to be better than others. I'm going to illustrate that with a website that allows me to change phi and ultimately psi and allow you to see what that does for the structure of the molecule. The bond in green is the bond between an amide nitrogen and an alpha carbon in black. Do you see that bond? Okay. So we're going to look down it in a Newman projection format, and I'm going to rotate around the bond between the nitrogen and the alpha carbon. And I've set this to illustrate anytime there are steric clashes. That means anytime things start to bump into each other. you can see that I can change phi a lot from around minus 57 or minus 50 to plus quite a bit without a lot of problems. But you see at the edge of that range, a yellow blob show up. And that yellow blob represents times when there are steric clashes. So here, for example, it's not the carbonyl carbon, but the carbonyl oxygen that's bumping into the side chain. This is what I meant by sometimes you have to think about things beyond just the atoms that are attached to the two atoms in the bond. That's a steric clash between the carbonyl carbon and the side chain. And at the other extreme, if we rotate back away, at the other extreme, now the carbonyl carbons start to bump into each other. So you'll see that phi has quite a bit of freedom, but it's constrained by steric clashes. All right, sorry, I wanted to do this. And I don't know why it zoomed out there. That was kind of weird. Questions about that? I've linked to this in your study guide. You can play around with it if you want, and it can be an effective way to learn what phi and psi mean. We're looking again down the bond between the nitrogen and the carbonyl carbon. And in general, you want to avoid steric clashes. You can see that changing the size of the side chain might change when these steric clashes start. In this amino acid, the side chain is just a methyl group, which is relatively small. If that side chain were bigger, there would be a chance for more steric clashes. Okay, now we need to talk about the next dihedral angle, which is the Greek letter psi. This is looking down the bond between the alpha carbon in front, sp3, and the carbonyl carbon in the back, sp2. The alpha carbon in the front is going to be bonded to a hydrogen, the side chain, and the amide nitrogen. The carbonyl carbon in the back is going to be bonded to the oxygen, and then the green amide nitrogen. We're looking down this bond, and here's what we would see in the Newman projection. In the front is the alpha carbon. Here's the proton, the side chain, and the amide nitrogen. That's our sp3 atom. In the back, you've got the carbonyl oxygen and the green amide nitrogen there, and that's a trigonal planar carbon. The angle psi refers to this angle, the dihedral angle between the nitrogen in the front and the nitrogen in the back. And in this alpha helix, that angle just happens to be around minus 50, about minus 47. And again, you can see that the carbonyl carbon oxygen is sort of eclipsed with the proton on the carbon in the front. But that allows the NH group to be somewhat staggered in between the side chain and the NH in the back. Okay, so that's the definition of psi. Again, we can go to our sort of model, and instead of, I'm going to reset phi back to where it was. We'll put it to sort of where it roughly is in an alpha helix, around minus 55. Then I'm going to switch to psi. Nope, I didn't want that. And that's the bond in green between the alpha carbon and the carbonyl carbon. Currently, psi is around plus 165. I'm going to show steric clashes. And you can see that psi actually can adopt values that phi can't. Psi is okay being positive. It starts to get some steric clashes around minus 135, but after that becomes pretty well okay. So psi has a little bit more freedom than phi does. Psi has fewer steric clashes. There you can see the amide proton in the back is bumping into the side chain, and the two carbonyl oxygens are bumping into each other. In general, you will see that that psi adopts a broader range of dihedral angles than phi adopts. If you play around with this website long enough, you'll see that the phi and psi values are interdependent. Somebody could ask me, okay, what if you changed phi by 20 or 40 degrees? How would that affect psi? And it turns out they do affect each other, and they are interdependent, and they do affect each other's steric clashes. Somebody has actually gone through and done the math and tested all possible values for phi and psi and showed where there are steric clashes and where there are not. And they've identified combinations of phi and psi that should be just fine without a lot of steric clashes versus combinations of phi and psi where there are steric clashes. Somebody else went on and measured actually the phi and psi angles for all proteins, and it turns out what we observe matches pretty closely with what the math predicts from trying to avoid steric clashes. And this data are summarized in what we will call a Ramachandran plot. So that looks horrible, doesn't it? Yeah. Let me take it over here where I can use a pointer. A Ramachandran plot measures phi and psi for each amino acid in a protein or a database of proteins, and then plots them individually as these little black dots. The phi values are on the x-axis. The psi values are on the y-axis. As you've seen, phi and psi can vary from 0 to minus 180 to plus 180. Each black dot in this plot represents an individual amino acid in a protein where somebody went and measured those dihedral angles. And what you see is there are areas of this plot, that is combinations of phi and psi values that are densely populated, where there's a lot of black dots, and there are places where there are almost none. These places where there are almost none, those are exactly the places where steric clashes happen and where they were calculated to happen. These areas where there are lots of examples are places where steric clashes don't happen as much. There are letters that label different portions of this plot, and that's by design. This portion is called alpha because if you see what an alpha helix is and then you measure the phi-psi angles in that alpha helix, most of them are here. If you do the same for a beta sheet, most of them are up here. And so you can be quantitative now on understanding protein structure in ways that you were only qualitative before. What is an alpha helix? Well, it's a thing that looks like a spiral. Okay. But it's also a chain of amino acids that each have phi-psi values in this range. Same thing with a beta sheet. What is a beta sheet? Well, it sort of looks like a zigzag. Yeah, but it's also a chain of amino acids where each amino acid has dihedral angles in this range. So you can use phi-psi values to be quantitative about what we call conformation or secondary structure in amino acids and proteins. Okay, questions? One thing somebody wants to know out there but is not willing to say is, do we have to memorize that? No. You should be able to understand and read it. For example, you need to know that phi is on the x-axis and psi is on the y-axis. You need to be able to interpret it. The fact that there's a lot of dots here means that that must be a combination of phi and psi that's okay and highly populated. And then given some data, you need to be able to draw conclusions. I won't say too much more about that, actually. In an alpha helix, in the thing that we just did, if you measured the phi and psi angles for the orange, purple, and green amino acids, they have these values, minus 55 and minus 45, minus 57, minus 47, minus 60, and minus 50. They all cluster in the same region of the Ramachandran plot, which I've sort of reproduced here, but you see compared there, that's what it means to be in an alpha helix. Okay. So how could you use these data to understand the relationship between sequence, structure, and function? We're going to talk about this by giving you some examples of the types of dihedral angles that are adopted by different amino acids. So beginning now, you should try to memorize, you need to memorize the names and structures of the 20 amino acids and their side chain pKa's as appropriate. We will talk through some of them, but some of them you're going to have to put just to devote some time to memorization. For now, don't worry so much about the side chain structures or the names. We're going to focus on how side chain structure influences conformation. So this is the amino acid alanine. That's the one we were looking at on that web page. And its side chain is a methyl group. Now, because the backbone is the same for all of the 20 amino acids, I'm just drawing a little squiggly mark with an alpha carbon there so you can know that this is what's attached to the alpha carbon. Understand that the stereochemistry is all the same at the alpha carbon, which is why I've not drawn it. They all look like this. We start counting from the alpha carbon, and the carbon that's attached to that is called the beta carbon. Now, hopefully that should be okay to you. For alanine, the beta carbon is a methyl group. Over here on the right is a Ramachandran plot for all of the alanine residues, all of the alanine amino acids or residues that were present in a protein database at the time the authors published this paper from which I've taken the structure. So phi on the x-axis, 0, plus 180, minus 180. Psi is on the y-axis, 0, plus 180, minus 180. One of the things you notice, again, like I told you before, looking at a Ramachandran plot, phi values are more restricted than psi values. Phi values tend to all be negative because phi suffers from more steric clashes. Psi values tend to be both positive and negative, much more flexible. The other thing you notice is this Ramachandran plot is not a plot of individual dots, it's a heat map. This is easier to interpret if the figure is pretty small. In a heat map, warmer colors represent higher population density, or in other words, the fact that it's red here means that there are tons of examples of alanine amino acids with that combination of dihedral angles. Blue areas of the plot represent fewer examples. So from this, we would conclude that alanine has tons of examples where phi is around minus 57 and psi is around minus 47, which is consistent with being in an alpha helix. You could conclude from this that alanine is pretty comfortable in an alpha helix. You only know that because there's tons of examples in the protein database where it is. In contrast, there are fewer examples, still a lot. Yellow is still a lot, but not as many as red, where alanine is in a beta sheet type structure. Okay? So what might I ask you to infer? Which conformation is alanine more comfortable in, alpha or beta? And you could say there's more examples where it's in alpha. Now let's talk about how side chain structure can influence things. I tried to show you earlier, or have you imagine what the impact would be of making the side chain here bigger. In valine, this beta carbon has 2 methyl groups attached to it, and you can see that that would make some of the steric clashes worse. So let's take a look at what that does to the Ramachandran plot. Here is the amino acid valine. Its beta carbon has 2 methyl groups attached. We call this valine beta branched because it's got 2 branching carbons on its beta carbon. Right away, you can see valine, like alanine, there's tons of examples in the alpha helical conformation, but there's also tons of examples in the beta sheet conformation. If you compare the plots for alanine versus valine, you could conclude that one effect of adding these beta branching methyl groups is to make valine more favorable than alanine in a beta sheet conformation. Okay, so side chain structure influencing what kinds of secondary structures that particular amino acid can adopt. So I could ask you, look at which of these 2 is more comfortable in a beta sheet conformation. You could say valine for sure. Okay, questions about that? Yeah.
So we need to memorize the name, the structure, and then you also want us to like kind of memorize what conformations are more.
No, I don't want you to memorize what conformations they can adopt, but I do want you to be able to use data to draw conclusions. So you're not going to memorize Ramachandran plots, but given them, could you draw conclusions? Okay, let's make a hypothesis. Supposing we replace the alanine side chain with a proton instead of a methyl group. What do you think that would do to the steric clashes that happen. Should decrease them, right? Then you might expect, if the side chain were a proton, that that particular amino acid could adopt a wider range of phi and psi values. Notice for alanine, there's nothing over here where phi is positive. We can actually go to that amino acid where the side chain is a proton. This is called glycine. I put it in a weird category of amino acids, and you see that it is. Unlike alanine, it's got tons of stuff going on over on the right-hand side of the plot. The reason is you've taken out a major source of steric clashes. So glycine, whereas alanine's only comfortable here and here, glycine's comfortable over here as well. This is sort of, glycine's Ramachandran plot is, the left-hand side is sort of an inverted reflection of the right-hand side. And it's because you've removed steric clashes. And we'll see in some proteins, perhaps we'll talk about an example of this next time, that sometimes you need an amino acid to adopt dihedral angles where phi is positive, and glycine is a good amino acid at that position and is often found there. Why? Because it lacks steric clashes so that it can adopt that dihedral angle. Yeah, go ahead.
Would it be presumptuous to say that the bigger side chain would mean fewer rotations?
Would it be accurate to say the bigger side chain maybe has more restricted rotation? That's a decent hypothesis, and it works, though it doesn't tend to matter much beyond the beta carbon. So you'll see that changes made to amino acid structure at the beta carbon change things quite a bit, the alanine-valine comparison. If we add things out beyond the beta carbon, it doesn't change it a lot relative to alanine. Okay, one other sort of example I wanted to do is to consider the polar amino acid serine. Serine is just like alanine, except it has an OH group. So we've replaced one of the protons on serine with an OH group. And you can see that serine, like alanine, is highly populated in the alpha region of the plot, maybe a bit more populated up in the beta region of the plot. Now, there's a close relative, a sibling to serine, that is going to have a methyl group at this beta carbon. That's called threonine. And so serine, threonine, like valine, is beta branched. It's going to have two things on the beta carbon instead of nothing or one. Before, we observed that adding the beta branching, alanine versus valine, increased the level of adopting a beta sheet-like conformation. Now make a prediction. What do you think adding the methyl group of the beta carbon should do for threonine versus serine? And a decent hypothesis based on data that you've seen before is maybe you ought to up the amount of beta sheet conformation. And here's the results. Here's threonine, and indeed quite intensely adopts conformations up in the beta region. This is an example of side chain structure influencing conformation. And remember, conformation or secondary structure is a prerequisite to function. So you're learning some of the language of protein folding now just based in side chain structure. And there's a lot more to talk about. One more weird amino acid that I'll say something about here just at the end is this one, proline. Proline is bizarre because proline has a carbonyl carbon, an alpha carbon, and an amide nitrogen, a beta carbon, but then the side chain is tied back on the nitrogen. This is the one and only amino acid where this is a thing. Because it's cyclic in this way, the nitrogen and the alpha carbon are not only bonded to each other, but constrained by a ring. You can see that that puts further constraints on what phi and psi can be. Compare the phi values for proline, which are very narrow, only in this sort of 50 to 60 range, versus alanine, where phi varies from minus 50 to like close to minus 180. Being part of a ring constrains the values that that phi angle can adopt. Okay, so that's your introduction to the relationship between sequence, structure, and function. Next time, we'll delve deeply into amino acid side chains and then start talking about protein folding.